Kamila Muchowska, profesora asistente del Laboratorio de Catálisis Química.
Hello, can everybody hear me? Can you hear me okay? Yes?
Yes,
okay.
We can hear you. Welcome again.
So I'm going to share the screen. There we go. Okay, you can see my slides.
Yes, we can see. Yes.
Excellent, perfect. So, buenos días a todos desde Estrasburgo, Francia. Antes de comenzar, me gustaría agradecer a los organizadores por darme esta fantástica oportunidad de presentarles nuestras investigaciones. Y también estoy muy emocionada de presentarme junto a científicos tan conocidos en este magno evento. Es verdaderamente una distinción muy grande para mí y por la que estoy muy agradecida. Gracias. And now I'm going to switch to English because I absolutely cannot talk science in Spanish. I'm really sorry about that. So before I get to science, I would like to show you a bit of Strasbourg, where I come from. Well, I don't come from Strasbourg, I work in Strasbourg. And Strasbourg is situated in the east of France, in the historic region of Alsace. So the entire historic city center is a UNESCO World Heritage Site. And Strasbourg is also the official seat of the European Parliament, which is one of the most important institutions of the European Union. And our Institute of Science and Supramolecular Engineering, where I work, is really a prime institution for multidisciplinary research that interfaces chemistry, physics, and biology. So I work in the Laboratory of Chemical Catalysis, which is directed by Professor Joseph Moran. Now, onto the science. But to do that, we need to go back in time by about 4.5 billion years. So 4.5 billion years ago, the Earth was very, very young, and certainly there was no life on it. However, thanks to the planetary scientists and geologists and many different scientific disciplines, we know that we have numerous models that can provide information about what was present on the lifeless planet Earth. And we roughly know when, for example, liquid water appeared. We know that the atmosphere of the planet likely contained lots of carbon dioxide. And we also know about the abundance of metal-containing minerals. So what in fact we did have on the early Earth is plenty of potential building blocks for the development of life. So perhaps my perspective here will be a bit different from the other talks at this conference, because I'm going to talk about the emergence of life on Earth and not about space. But I believe that some fundamental aspects that I will present will have a universal value for both the Earth and also other planets. So the general starting point here is that to have a deep understanding of life's chemistry, we also need to understand the chemical foundations of living cells. So we need to understand how they came to be. So my little disclaimer here is that I'm going to talk about life as we know it. And the model environment will be based on what we know about the early Earth. So in other words, the question we are trying to answer is, how did geochemistry here get organized, self-organized to become biochemistry, which you have here, and eventually biology. So this is how we currently depict biochemistry. This is an incredibly complex map. It contains chemical transformations which describe everything that keeps life alive. So two important things about this map is that it is very well organized. It contains patterns, you can see, like circles, kind of snail shapes. And another thing is that this map is not static. It is a dynamic network. All the time there are changes, there are fluxes. So this metabolic map is indeed very complex, and it would be hard to envision something like this organizing right away from the molecules on the early Earth. So this is why I'm showing you here a simplified version of metabolism. So it contains important subclasses of different biochemical, of this giant biochemical network. And so what this simplified biochemistry does is it takes CO2 here, it fixes it into higher carbon molecules. So here we have what we know as the Krebs cycle in biology. So it can run, different organisms can run in different directions. You can also call it the TCA cycle or the reductive TCA cycle, so the RTCA cycle. If it runs in the reductive direction, so counterclockwise, it builds up biomolecules. So from here, we can also go towards, for example, sugars. We can build up sugars, we can break down sugars, or we can add nitrogen and build up amino acids, go later towards more complex structures like proteins, peptides. Eventually, we reach ribonucleotides, RNA, and the genetic code. So this is the fundamental organization of the chemical reactions used by life. Over the past decade, the consensus amongst chemists has been that to solve the origin of life problem, we need to find a good way to make biomolecules from simple substrates. So the major part of prebiotic chemistry, so the origin of life chemistry, of this research, it has focused on making the biological building blocks, such as amino acids, nucleobases, and so on. So in this slide, you see some examples. These are really classical examples of prebiotic chemistry. And what they're based on is usually hydrogen cyanide. So they are heavily based on hydrogen cyanide chemistry. These are really beautiful examples of organic chemistry. And I'm not an organic chemist, but I still can really appreciate the chemistry that's going on here. But I must tell you that if you ask a biologist, they will probably say that these intermediates look kind of foreign to them, and they are not really what biology uses to make these biomolecules. The biochemical pathways are quite different. They use different mechanisms. So now I would like to bring to your attention a different approach. So to do that, let's consider the organization of life, which follows the order of complexity. So this seems logical at the first sight, but also from the origin of life perspective, there is a massive chicken or egg problem. So in biology, metabolites are produced in reactions that are catalyzed by enzymes. Enzymes are complex proteins, and how they are made is encoded in genetics. But genetics, the genetic material, is also made from metabolites. So how do we break out of this cycle? So to do that, we need to assume that at life's beginnings, metabolites, which are here, they must have been produced from environmental inputs. So on the other hand, now it also becomes more evident than ever that making life's building blocks alone, this is not everything to solve the origin of life problem. This is not enough. Why is that? Life is a disequilibrated open system, and it follows the same rules of organization as, for example, a hurricane. So here on the left-hand side, you have a hurricane, and the building blocks of a hurricane are air and water. But you can put air and water together, even you can stir it like this, and you will not obtain a hurricane, because to do that, you need to apply an energetic stress over an appropriate scale and appropriate amount of energetic stress. And as a result, you obtain a process, you obtain a dynamic process, which we call wind. And the same thing happens with life, the same rules apply. We know the building blocks of life quite well, CHNOPs, elements, metals, small molecules, and in the same fashion, if you take all the necessary biomolecules and add them together, apply some stirring, you will not obtain life. That would be magic and not really reality. The point is that some disequilibrium in the environment of the early planet was absolutely necessary for chemical reactions to organize themselves into a process. And this process we know today as metabolic pathways. So how to generally approach this? Again, in a living organism, metabolic processes are generally enabled by complex proteinic enzymes, like this one here. You've got a beautiful enzyme. So often these processes consume ATP, an energy currency. If you want to do something, oftentimes you have to pay in these molecule, a high energy molecule, a complex molecule that was not present on Earth. So what I want to show you here is an approach to the origin of life on Earth problem. How we approach it is we draw a parallel between the general way that biochemistry operates in cells and how it might have operated very early on when the enzymatic machinery, like molecules like ATP, had not yet evolved. But as we know, there were much simpler catalysts available in the environment, metals, metal-containing minerals. Now, given how complex biochemistry is, we saw the giant complex map. How do we choose which pathways we want to work with first? To help us understand which kind of biochemistry could have originated as prebiotic chemistry, so before ATP, before enzymes, it can be helpful to make two fundamental distinctions within life. So the first one is whether a metabolism is reductive or oxidative. Does it get the energy by taking electrons from the environment, or does it get the energy by giving away electrons to the environment? Another distinction is the one between autotrophs and heterotrophs, where autotrophs contain the entire metabolism within a single organism, and heterotrophs share it with other organisms, which means, for example, they have to eat other organisms to survive. On the early Earth, we can be quite sure there were not so many organisms inhabiting a niche, and the environment, according to many, if not most models, was very mildly reducing. So many evolutionary biologists and chemists, like these guys here on the right-hand side, usually consider reductive autotrophs as the starting point, although there are probably other scientists who may disagree. Anyway, let's see what information we can extract from reductive autotrophic metabolisms. There are 6 or 7, depending on how you count, carbon fixation pathways that we know of that are used by microorganisms to build biomolecules. From the evolutionary standpoint, 2 of them are the most relevant, and I'll show them here. So the first one is called the Wood-Jungdal pathway, or another different name is the acetyl-CoA pathway. It is short, it is linear, it produces more energy than it consumes, so it's a downhill pathway. It is found at the very, very root of the tree of life in bacteria and archaea. And it is also thought to have been present in the last universal common ancestor to all life on Earth. So it takes 2 molecules of carbon dioxide. If you're not a chemist, don't worry. I will have chemical structures, but I will walk you through it and I will explain what's going on here. So at the end of this pathway, you obtain a molecule that has 2 carbons. So from 1, you go to 2. Another pathway that's relevant is the reductive Krebs cycle and the reverse Krebs cycle. So you start from here, acetyl, and you build up biomolecules. You keep adding carbons, and eventually you end up with a 6-carbon molecule of citrate, which then becomes broken into 2. So this cycle is quite interesting because of several reasons. One of those reasons is that it contains 5 universal metabolic precursors to different classes of metabolism. So, for example, from the acetyl molecule here, you can synthesize lipids, another pathway. From pyruvate, you can go towards amino acids, alanine, also towards sugars. This will be important for later. From oxaloacetate, you can go towards amino acids and pyrimidines for the synthesis of ribonucleotides. From glutamine, you can go towards amino acids. From succinate, you can go towards pyrols and different cofactors, which are also important. So that's one thing. Another thing is that this cycle, with every turn, it self-amplifies because you have this loop here. So you run it one time, you get 2 of these structures. You run it another time, you get 4, then you get 8, and it's an exponential growth. So it's really quite interesting, this topology. So it has been hypothesized that these 2 pathways were once fused, and they worked together in the prebiotic world. So it would have looked like this. So then we, in our lab, we wondered if we can obtain such a network without enzymes, how would it function? On one hand, chemists could think that this is a bit unlikely to work because CO2 in water is not very reactive. But we also saw that many of these reactions in biology use enzymes which contain transition metals in their active sites. So we've got nickel, iron, manganese, magnesium, cobalt, molybdenum, tungsten, many different metals. And transition metals are also used all the time as catalysts in organic synthesis. So the question was now, could metals alone do exactly the same job and promote these reactions in some way? So we organized ourselves some pressure reactors to be able to work with gaseous CO2, and we started the experiments. So we did some high throughput screening with different metal catalysts to see if any of the reactions of the acetyl-CoA pathway could be promoted this way. So we were especially curious about iron, because iron is geochemically the most abundant metal and arguably also the most important one in biochemistry. So you've got the CO2 metal catalyst, and we looked for acetate and pyruvate, which have 2 carbons and 3 carbons respectively. So it turns out that if we take the metals that we find also in the acetyl-CoA pathway, in the enzymes, so molybdenum, iron, nickel, cobalt, manganese, and tungsten, we place them in a powder form in salty water in little vials, and we expose these vials to carbon dioxide at 100 degrees Celsius. It turns out that we can fix carbon dioxide into biomolecules, so acetate from 2 molecules of CO2, and even pyruvate from 3 molecules of CO2 in the case of iron, of nickel, and of cobalt. So this simple reaction directly follows the intermediates of the first carbon fixation pathway that I showed you just a few slides ago. We probed this chemistry further for iron because it was an interesting metal for us, the most abundant, most important. So in this experiment A, we looked at the effect of temperature. And it turns out that this reaction works all the way down to 30 degrees Celsius, which is lower than body temperature. And at 30 degrees Celsius, it's also more selective. So it actually gets worse when you get to higher temperatures. And 150, there is no more pyruvate because we think it decomposes at this temperature. In this experiment B, we probed the effect of pressure. And it turns out you don't really need a high pressure of CO2. It works at 1 atmosphere of CO2. And the reaction becomes more selective, and acetate and pyruvate are the only products. So we were quite astonished to find this. And we also monitored how these reactions change over time. There is a lot of interesting information here, but we don't really know what it means. It's quite complicated, this heterogeneous catalysis, this chemistry. So we didn't really know how to exactly interpret these results. At the end, you start seeing even a bit of ethanol in some cases. And recently, we established a really nice collaboration with our collaborators in Germany and in Japan. We all got together and found that you don't even need metallic iron to promote the acetyl-CoA pathway without enzymes, because it works perfectly well with hydrogen gas and iron and nickel containing hydrothermal minerals if you use them as catalysts. So these minerals you can find on Earth. These are natural. So this way, the potentially prebiotic carbon fixation pathway becomes really similar to the biological pathway, because biology also uses hydrogen in its analogous process. So, can primordial metabolism work without enzymes? The answer is yes, at least for this part of the network. So let's go on then to the next part. So this is the reductive Krebs cycle, which we also call the RTCA cycle. It runs in the anti-clockwise direction. Starting from here, you build up higher carbon molecules. So there are 11 steps in this pathway, but only 5 types of reactions. So 2 types of carboxylation, reductions, hydrations, dehydrations, and retroaldol cleavage. So 5 of these steps require energy from ATP in biology, and 6 of them do not. We were clearly not the first ones to try to recreate the RTCA cycle reactions under possibly prebiotic conditions. So, for example, Scott Martin and co-workers used the photocatalytic properties of semiconducting minerals like zinc sulfide and manganese sulfide to catalyze 2 of the reductions of the Krebs cycle, of the reductive Krebs cycle, using UV light and electrons from H2S. But unfortunately, only 2 reductions here gave yields over 3%. They were not very efficient, and no continuous sequences were obtained here either. So our work on the RTCA cycle, we started also from the irreversible reductions, which are the pink blobs here. But we also coupled them to other reactions with the hydrations and dehydrations to obtain entire sequences. So we tested different conditions, temperatures, pH values, different reducing agents, different metal salts, different additives. After about 4,000 reactions, we found that metallic iron and zinc salts could uniquely promote not just a single step, but entire sequences in one pot under acidic conditions. So basically, you take oxaloacetate, this molecule, apply these conditions here, wait a little bit, heat it, and at the end, you end up with this one. So that's the bottom arc. So for the upper arc, we started from oxalosuccinate. We found that the addition of zinc salts and chromium salts can promote this upper sequence in one pot. So basically, you stick the oxalosuccinate molecule in your reaction mixture, add the salts, add the reducing agent, wait, and at the end, you get a mixture of these intermediates and citrate. So the both arches, they work in under conditions that are mutually compatible, and they are compatible with each other. So we were quite happy to find that. And these were all steps that are ATP independent in biology. Now I want to show, especially to non-chemists, what it looks like when I talk about high throughput screening. So we run dozens of reactions at a time in small vials, and by changing parameters such as temperature or different additives or pH values, we determine the optimal conditions. So we have, our laboratory has a lot of experience in dealing with high throughput screening and complex mixtures. So most of these reactions were analyzed by gas chromatography coupled to mass spectrometry, so GCMS, and also by NMR spectroscopy, such as this machine here. So to recap quickly on where we are, after screening for both pathways, it was roughly 10,000 reactions. So we found that the combination of metallic iron and transition metal salts could promote all of the ATP independent steps of both pathways in continuous sequences, and that's important. However, the problem here was that we weren't able to promote any of the steps that are dependent on ATP in biology. And these are the ones that you have highlighted with pink blobs with ATP. ITP is here. So most of these steps that require ATP, these are carbon-carbon bond forming reactions. So to build a carbon skeleton, to add more carbons, to make a more complex molecule. And this is really necessary for anabolic processes to build up more complexity. So, so far, no ATP-dependent CC bond-forming reactions have been shown to have prebiotic analogs. And it is really unclear whether it's because it's difficult, or is it something about kinetics, or because this is a new approach to prebiotic chemistry and not so many people have tried to have a go. So how did life ever come anywhere near building carbon-carbon bonds without ATP? One important hint came from a paper by Segre from 2017, from a 2017 paper. So in this study, what they did, they took the entire library, the KEGG database, and this is a library of all known chemical reactions. So they took it and they performed a computational study where they removed from this library all the reactions that contain any sort of phosphate. ATP contains phosphate, so they also removed ATP. So what they got is something like this. If you have no idea what you're looking at, try to imagine this is a subway map. So you've got different lines, different trains, and the nodes here, they are stations. So if a station is very busy and has a lot of traffic, it will be bigger. So in the same way, it works here. The most well-connected nodes here in red, this is pyruvate and glyoxylate, two important biomolecules. So this metabolism here, it's hypothetical. It's a hypothesis. So what they obtained is something that does not require phosphate, and it is still quite well connected. You have many reactions that you don't have really islands of isolated compounds. They are all very well connected. So if this metabolism, hypothetical one, ever existed or ever functioned, it would have heavily relied on these two hub molecules, pyruvate and glyoxylate. So this is where you can find them in live rotten apples and fresh apples. A bit later, a paper came out by Springsteen and Krishnamurthy and co-workers, and it really reinforced the role of these two simple biomolecules in potential protometabolism. So they performed a bottom-up or chemistry-up approach study, and they reported an oxidative decarboxylation-driven bicyclic network of two cycles, seeded by the aldol reactions of glyoxylate and pyruvate, oxaloacetate or malonate. So in the presence of hydrogen peroxide added as oxidant, they obtained these two cycles. So the net result of these experiments is the formation of a reaction network with this nice bicyclic structure, and oxaloacetate is a pivotal link, and malate and oxaloacetate are, malate is here, oxaloacetate here. These are both intermediates of the Krebs cycle. So these are biological molecules, really important. Last year, we tried to do a similar thing, except quite differently, because we took pyruvate, we took glyoxylate, and we added metals, we added iron. So this is what happens. If you add pyruvate and glyoxylate together, you add iron, 70 degrees Celsius in water, and you wait. And this is what happens. Initially, nothing really, but as time goes by, you obtain 16 different biological compounds after 24 hours. You don't have to know what they are. These are all biological compounds. Most of them belong to the Krebs cycle, actually. So there is a lot of complexity growing. At the same time, this graph shows the mass balance. So you're also losing the mass. The longer you wait, the more complexity you obtain, the more mass you actually lose. Where does it all go? Back to CO2. And this is actually a good thing, and I will explain that in a moment. So this is the network that we obtained for chemists now. So you start from here, glyoxylate and pyruvate, mix them together in the presence of iron and warm water. And then as time goes by, you obtain more and more biomolecules. You obtain this sort of dynamic network before it collapses back to CO2. So finally, if you introduce a source of nitrogen in the form of hydroxylamine, you can obtain 4 amino acids, biological amino acids, directly from this network. So glutamic acid, glycine, alanine, and aspartic acid. To give you a better idea of what I'm talking about when I mentioned that all these molecules belong to the Krebs cycle and so on, it's hard to imagine. So here is the same network as on the previous slide, but it is redrawn and overlaid over 2 biological carbon metabolism cycles. So here in red, you have the Krebs cycle on the left-hand side, and another one on the right-hand side is a glyoxylate cycle. This is another biological pathway present in plants. So it turns out that our network contains 9 out of 11 intermediates of the biological Krebs cycle, and it also recapitulates 7 out of 11 reactions. And the same goes from the glyoxylate cycle, 8 out of 9 intermediates, the fat arrows, they show what we have in our network, 5 out of 8 reactions. So we are really starting to recreate biochemistry without enzymes. And if there are no enzymes, this is a biochemistry that operates driven by an inorganic promoter instead of an enzyme. Now I said before that it's actually not so bad that the intermediates of the network break down to CO2. And this is because life builds up molecules and breaks them down in a steady state. Just like this fountain in Strasbourg pumps up water, it goes up, and then it falls back down under gravity. So the goal of our study was to demonstrate an abiotic system that could show just that molecules are made from CO2, they can react to build up complexity, at the same time they break down again back to CO2. So we wanted to show that this can happen in ways that are similar to biological pathways with minimum human interference. So the point was to draw the analogy with planetary and biological carbon cycles. It is completely possible that anabolic and catabolic processes, so the build-up and the breakdown, needed to be linked from the very beginnings of prebiotic chemistry. So in other words, what this means for us is that non-enzymatic proto-metabolism would have necessarily emerged in a formation-destruction regime. That's what we call it. There would have been a requirement for available electron donors and acceptors, and the system would have needed to be part of, for instance, a geochemical redox cycle. And the building blocks, they would also have needed to be constantly replenished. So the whole system can remain in a disequilibrated state. So again, this is we are back to the big picture. We've got the acetyl-CoA pathway functioning so far, the RTCA cycle, some amino acids. We've got the network that covers most of the TCA or the RTCA cycle. Where do the breakdown processes fit, and how do we achieve them? So actually, several of them have already been reported. And this is an example of a study by Marcus Ralze and co-workers. And they showed actually that the oxidative Krebs cycle can be promoted with peroxydisulfide, disulfate with under iron catalysis at 70 degrees. So look at these conditions. They look quite similar to what we had, right? They look familiar. And in another 2 papers by Ralze, the same scientist, they showed that 2 examples of the breakdown of sugars. So they showed an analog of biological glycolysis, and they showed an analog of the biological pentose phosphate pathway. Also under the conditions that are quite familiar to us. So you go from glucose down these pathways to end up with pyruvate And in the meantime, you obtain also all these intermediates here. These are it doesn't really matter what they are at the moment. But you could also ask if, aren't these breakdown processes just kind of pointless? Because it seems like a waste, right? It's a complete waste. And the answer is no, they are not pointless, as long as the breakdown products can be trapped into something useful. And biology does exactly that. So this is a simplified biological Krebs cycle. So in some of the steps, it uses thioester. Thioesters. It has some of thioester-dependent steps. And these are highlighted here in yellow. So generally, if the Krebs cycle, if you lose a molecule of CO2, the rest is trapped into a thioester, which is then involved in the phosphorylation of ADP to ATP. So these are very important steps in the Krebs cycle. The thioesters used here, they involve a coenzyme A. So coenzyme A is here, a huge molecule, complicated, contains phosphate. Yeah. So in order to not have to work with this complex molecule, which was quite, it is quite certain that it was not present on the early Earth, it's too complex, we took the N-acetylcysteamine. So this is the red tail with a tile function. And we tried to see if we can obtain a similar reactivity, the same type of chemistry within the breakdown processes in the pyruvate glyoxylate network. So these results here, they were obtained by my colleagues, Dr. Jan Gorges and Dr. Elodie Chevalot-Beroux, who worked in our group. They found two types of conditions, thermal and photochemical. Under these conditions, you can obtain thioesters from alpha keto acids. So here, thioesters in yellow, yellow, yellow. And the rest of the molecules, they are the complex network that I showed you a couple of slides ago. So the conditions that they found, also quite interestingly, peroxide disulfide, and iron catalysis. So quite similar to what I showed you before in other studies. So it's kind of converging on a type of conditions. If you hydrolyze these thioesters, you get rid of the thiol, you get back to the regular carboxylic acid, just like in the Krebs cycle. So hydrolysis for now of these compounds is quite sluggish, happens super slowly, but if it happens, you obtain the molecules that are also on the cycle. So now, what do we have left? This is pretty much the end of what we have reported so far, but I would also like to tell you what the Moran Lab is working on at the moment, and also where we want to venture as part of our future work. So that is expanding the non-enzymatic prebiotic chemistry towards sugars, here in blue, and also ribonucleotides, amongst other metabolic subsets. So one popular approach in origin of life chemistry to sugar-forming reactions involves the formose reaction of formaldehyde, but it leads to a very complex mixture of pentoses, hexoses, and essentially at the end you end up with tar, and it's not selective at all under most conditions. And another popular approach is the so-called Kiliani-Fischer homologation of hydrogen cyanide. I'm now not going to delve into this in much detail, but instead I will show you the biological pathway. So this is the gluconeogenesis, and it starts from a Krebs cycle intermediate, pyruvate. So then you have phosphorylations, dephosphorylations, you have an aldo reaction, and you end up with glucose. So this research is actually currently pursued by my colleagues in the Moran Lab, Dr. Ankita Malik and Joris Zimmermann, and also our stagiaire, the intern, Elodie Didier, who left recently. And we actually have some exciting results, but I'm afraid I can't really share them just yet, but stay tuned for updates, follow us on social media. We might have something really interesting, really cool to show soon. And of course, here is the reminder that the conceptually reverse pathway, so the breakdown process, catabolism, glycolysis starting from glucose, or the other pathway called the pentose phosphate pathway, they have already been reported. And the important molecule that they produce is this one. This is ribose 5-phosphate. So why do we need it? Ribonucleotides, finally. So the general skeleton of ribonucleotides is based on the ribose 5-phosphate sugar units, here, blue ribbon, and 4 nucleobases, pyrimidines, uracil and cytosine, and purines, adenine and guanine. And they also have to be made somehow. So again, the classical approaches are heavily based on hydrogen cyanide, on the nitrile chemistry. And over the past few years, we have been able to witness really fantastic organic chemistry works, proposing pathways towards ribonucleotides involving nitrile cyanide intermediates, like this paper by Sutherland from 2009, or more recent from Science by Carell and co-workers. However, hydrogen cyanide is not used by life to make nucleobases. So instead, let's have a look at what biology does. In biology, both purine and pyrimidine nucleobases are built from metabolites. The C1 units, blue, dark blue, are supplied from formate, this molecule, or bicarbonate, the light blue. And a large part of the carbon skeleton, as well as the nitrogen involved in these structures, they're supplied by amino acids, such as glutamine, glycine, and aspartate. And these amino acids can all be obtained from Krebs cycle intermediates. And indeed, several of my colleagues in the Moran lab have taken on the biosynthetic pathway of pyrimidine ribonucleotide synthesis. So they're trying to propose its prebiotic non-enzymatic analog that follows the biological pathway. And they also obtained some really exciting results that I cannot, again, tell you about just today. But please stay tuned for updates. It will be very exciting for us to share the new research, the new results, maybe next time. This brings me to the end of my talk. Of course, this simplified biochemistry you can see here is largely based on the central carbon metabolism. And it's obviously only just a small part of the huge complex metabolic map. So, and this metabolic map also, it only describes only what we know about life as we know it, life on Earth. But obviously, it's also a great point for further explorations. So what I would like to emphasize here and recapitulate the point that I made with the hurricane slide is that in living organisms, metabolic pathways need to work together as a whole. Nothing is really a standalone chemical system that works disconnected from everything else. So what this means is that any constraints that we apply to non-enzymatic chemistry of one reaction of path or one pathway become also relevant to all the other pathways that we want to work with. Because all these subsystems that we see, different classes of biomolecules, they remain kinetically coupled in one way or another. So ideally, in prebiotic chemistry, they must be all considered simultaneously. You have to always keep them at the back of your mind. If you work with ribonucleotides, keep in mind that there's also gluconeogenesis that has to be kind of connected conceptually. So this is really important when you're drafting a prebiotic scenario. What remains to be thought about as part of future work is this sort of transition. Let me explain. We know that some of the metabolic pathways that I showed you, they can be promoted by simple catalysts from the environment, so rocks, for example. But how do we get from metal-containing rocks to here, this complex metal-containing enzyme, proteinic enzyme? So surely there must have been something in between, but we don't know what it is. And this is where my current research is trying to fill the gap. I'm focusing on two new and two complementary aspects. One of them is looking at out of equilibrium systems by using flow chemistry. And another is trying to explore the chemical evolution of cofactors, such as this complex molecule of coenzyme A, because cofactors might have been involved in the earliest version of metabolism, before the emergence of enzymes, which are even more and more and more and more complex than the molecule that you can see here. Unfortunately, little is known about the prebiotic synthesis of cofactors in a way, at least in a way that would recapitulate biosynthesis. And even less is known about the functional rationale behind the structure. Why does coenzyme A look the way it does? Like, why? These projects are ongoing at the moment, and I will be really excited to share some results with you next time. To summarize, the main conclusion I would like to make in this talk is that non-enzymatic analogs of the core metabolic pathways may have predated the biological enzymatic ones. Secondly, these non-enzymatic protometabolic pathways can be promoted by simple catalysts that were likely available on the young planet. However, there is still a great number of really fundamental questions about the nature of life, and I admit that we are only just beginning to scratch the surface of it. So while we do this, it's important also to remember that life itself is a fantastic guide to uncovering its own origins. We should look at life and learn from it, learn from what life does. And exploring biochemistry in the context of the origin of life, it will definitely make for exciting years of work ahead of us. I would like to thank a bunch of people now, because I'm just talking about research, but none of this has been done single-handedly. And there is an entire team and really many people who have been involved in this research, very fantastic people. So first of all, I would like to thank Professor Joseph Moran, who is the director of our lab of Laboratory of Chemical Catalysis at ISIS in Strasbourg. A fantastic mentor for me, so thank you. And also Srijit Varma, who has worked with me over several years as a postdoc, and who took part in a large part, majority, I would say, of the results that I showed you today. And also other people involved in the Origin of Life project. We have a very international group, a very international institute. I would like to thank our collaborators in Germany, and also the funding bodies who have contributed to our research by providing funds. And I also thank you for your kind attention. I will be happy to answer questions. Thank you.
Thank you very much, Dr. Muchowska. I have some questions from creo que no me escucha. Do you listen to me? She's not listening.
Thank you, Dr. Kamila. We have time for 2 or 3 questions.
Do you listen? Okay.
Yes, I can hear you.
Okay. Sorry. I want to thank you for your intervention. It's very interesting, everything you shared with us. We had some questions from some people. The first is, the metabolic networks of eukaryotic organisms are very complex. Is it possible that some of these networks have been formed in different organisms, and then, by evolution and chemical selection, have been integrated by common metabolites?
Well, really interesting question. There are several theories that talk about the evolution of metabolic pathways. We have recently published a review on that in Chemical Reviews. where we try to explain some of these theories, some of these hypotheses in an accessible manner. It's hard to say. Well, I would like to say yes. It doesn't sound impossible to me. It is not so simple to explain how the metabolic pathways might have evolved, because you always have very different processes, such as the horizontal gene transfer and processes like that are involved in the evolution of the final version of metabolic pathways. On the other hand, if you have a pathway which is a part of a more complex system, it becomes kind of locked, because if you have a pathway that is in the center, then you have a lot of other pathways evolving around it. If you want to change the central one, it will trigger change in all the other ones. This is called a frozen metabolic accident. So I don't want to say no, but I can't really say yes for sure. I would lean towards yes in this question. It's possible.
Very much. Another question is the environment of the primitive Earth was different from the present. How are these conditions recreated in the laboratory, and what possible errors can variables cause?
Another very interesting question. So obviously we cannot go back in time and check what the environment on the early Earth was, but we learned from models of the environment. So there are different models. Some people say that you can find a scientist who will find you a model that fits your experiments, but I don't want to do that. I would say the consensus is that the environment was neutral-ish to mildly reducing, so we know about that. We also know roughly about the temperatures that could potentially harbor life, that could be conducive to life emergence. We know more or less what gases were present. So if we know these variables, we can recreate these conditions in a laboratory. So the temperatures that are kind of mild between room temperature and 100 degrees. So that's where I would say we roughly operate. Also CO2 as gas, the atmosphere, hydrogen gas also. So and we also know what sort of rocks and what sort of chemical composition of rocks was available on Earth. So we can also recreate this quite easily in the lab. But still, it's a non-falsifiable hypothesis. You cannot go back and check.
Thank you very much. We have a last question. Do you think that by introducing organic molecules with functional groups similar to the exposed cycles, but which are not on Earth, but have been found in carbonaceous contrites, the cycles can work and thus have extraterrestrial links molecules that have already been lost from the cycles of the life?
Oh, wow. This is a really tough question. Well, okay, I will try to answer in two ways. First of all, if you are wondering whether this type of chemistry that I showed you could function somewhere else, for example, in meteorites, if the conditions were similar, or on other celestial bodies like moons or planets, then I would say absolutely yes. Because if you have the environments, if you have the chemicals, then why wouldn't it work? If some molecules could have been removed from the cycles, I'm not sure if I understand the question correctly. Could you read the last part again, please?
The last part you say. From where?
It could be delivered to Earth by carbonaceous meteorites.
The cycles can work and thus have extraterrestrial links molecules that have already been lost from the cycles of the life?
I'm not sure if I understand this correctly, but if the question is about whether these sort of cycles and molecules could have been delivered by extraterrestrial means, so for example, meteorites to Earth, if this could have kickstarted life on Earth, I would say no. Well, yes, they could have been delivered, but if we are relying on this sort of delivery of any sort of chemistry to Earth to kickstart life on Earth, I would say no, because this is relying too much on chance events. And my personal opinion is no.
Thank you very much. Then we thank you a lot for being part of this event. We are very grateful for your intervention, for answering the questions of the people who are watching this event. And we want to ask you to send us a message to the people who are watching this here in the university.
I thank you for the invitation. It's been a pleasure. I'm really honored to be able to present amongst really amazing speakers. If there are more questions from the audience that I have not managed to answer, please do not hesitate to reach out by email, or by social media. I will be happy to talk and to answer questions. So thank you very much
for sharing with us in the name of the Universidad del Atlántico. We thank you very much for being part of this activity.
Thanks. So I disconnect now, right?